Hello class, welcome to part 2 of your digestive system installments. This slide I'm going to cover the histology of the alimentary canal wall, and I will move from the external surface to the internal surface or luminal side. That would be the side that faces the food that you just ate. If I'm going from the external surface to the internal surface, I'm going to start with the serosa versus adventtia, then continue deeper to the muscularis externa, deeper still to the submucosa and its components, and then lastly the mucosa. The outermost tunic may be either an adventtia or a serosa. An adventtia is composed of areolar connective tissue with dispersed collagen and elastic fibers. A serosa has the same components as the adventtia, but it is covered by a visceral peritoneum. Intraperitoneal organs have a serosa because they are completely surrounded by the visceral peritoneum, of course. Retroperitoneal organs primarily have an adventtia because these organs are only partially covered by visceral peritoneum. For example, the ascending colon, which is retroperitoneal, has an adventtia, whereas the stomach, which is intraperitoneal, has a serosa. Going deeper into the alimentary canal wall, I now get to the muscularis externi layers. There are two layers, the outer longitudinal layer and then an inner circular layer. The fibers of the outer layer are oriented lengthwise along the GI tract. The inner circular layer, the fibers are oriented in a circumference around the GI tube. In between these two layers is a blanket of nervous tissue, or a plexus, and this is called the myenteric plexus. These nerve fibers and the associated ganglia are going to help these two layers of smooth muscle with their contractions. It is important to note that this myenteric or Auerbach's plexus is the enteric nervous system. It's completely contained within the gut. It's strange to think that the gut has its own nervous system, but that's what it has. It has as many neurons as your central nervous system, brain and spinal cord combined. Now we're going deeper to the submucosal layer. The submucosa is composed of either areolar or dense irregular connective tissue, and it has fewer cells than the lamina propria, which I will talk about in a little bit. It consists of lymphatic tissue. It also has mucin-secreting glands that project ducts across the mucosa and then in turn open into the lumen of the tract, particularly in the esophagus and duodenum. Many blood vessels, lymphatic vessels, and nerves are found in this layer. In particular, the submucosal nerve plexus or Meissner plexus. These nerves are going to provide sensory information and control the release of digestive enzymes from the intestinal wall, as well as mucin secretion. We will see that in both the submucosal nerve plexus as well as the myenteric nerve plexus have both sympathetic and parasympathetic influences. There's a difference between the intrinsic nervous system of the digestive tract versus extrinsic influences, and I will continue this on the next slide. Going deeper towards the lumen, now we get to the mucosae, and the mucosae consists of three things. It has an inner superficial epithelium, that would be the luminal side. It has an underlying areolar connective tissue called the lamina propria, and then it has a thin layer of smooth muscle called the muscularis mucosae. Now this muscularis mucosae is not a strong band of smooth muscle. It only causes local contraction. I like to think of it as if there was a piece of potato chip stuck in the epithelium in this picture on your screen. The muscularis mucosae is just strong enough just to flick it back into the lumen to allow for proper digestion. Again, this is a picture for the layers of the gut. Going from the top of the screen down, we're going from the luminal side of the gut to the external surface of the gut that would face the peritoneal cavity. Again, we have the mucosa with 3 layers, the epithelium. Now, the epithelial layer is going to change as we go through the digestive tract. For example, in the esophagus, it's stratified squamous epithelium, but once we get to the stomach, through the large intestine, it's going to be simple columnar epithelium. When you get to the anus, it'll transition back to stratified squamous epithelium. That epithelial layer has to be anchored to the underlying layers by a lamina propria, or a sheet of connective tissue, in this case, areolar connective tissue. And then we have the muscularis mucosae. Then we have the submucosal layer. In the submucosal layer, we're going to see lymphatic vessels, blood vessels, and glands. And the submucosal nerve plexus that is found there is going to control these functions. It'll control release of secretions. It also relays sensory information. You do get information about the food that you eat and whether or not it's going to, quote, agree with you or not. That submucosal nerve plexus is enough to provoke increased gut motility. It can communicate with the myenteric nerve plexus found in between the 2 muscularis externi layers. Again, in this picture, you see an inner circular layer and an outer longitudinal layer. In between them, you see Aurobox nerve plexus, or the myenteric plexus. And then finally, in this picture, we're seeing the serosa. This picture is not in your book, but I really want to emphasize the gastrointestinal tract has its own nervous system called the enteric nervous system. That's shown here in the picture as the myenteric plexus and submucosal plexus. These neurons lie entirely in the wall of the gut, and they begin in the esophagus and extend all the way to the anus. The 2 plexi again are the myenteric and submucosal plexus. I want you to understand that the enteric nervous system is its own system of nerves. It has its own sensory neurons and visceral motor fibers, and as such, there are gastrointestinal reflexes that don't require the central nervous system. These enteric reflexes control gastrointestinal secretion, peristalsis, segmentation, etc., just to name a few. The myenteric plexus, or Aurobox plexus, is an outer plexus located between the muscularis externi layers. Stimulation causes increased tone of the gut wall, increased intensity of rhythmical contractions, increased rate of contraction, and increased velocity of conduction. The myenteric plexus is also useful for inhibiting the pyloric sphincter and ileocecal valve, which I will show you later on in this PowerPoint. The submucosal or Meissner's plexus is an inner plexus that lies in the submucosa. It is mainly concerned with controlling function in the inner wall of each minute segment of the intestine for local control of intestinal secretion, absorption, and contraction of the submucosal muscle, meaning the muscularis mucosae. There are parasympathetic and sympathetic fibers that synapse with the enteric nervous system, as shown in this picture. These can merely modify the activity of the enteric nerves. Yet again, the emphasis is on the word modify, as the enteric nervous system controls most of its own activity. The esophagus is a tubular passageway for swallowing materials being conducted from the pharynx to the stomach. The inferior region of the esophagus connects to the stomach by passing through an opening in the diaphragm called the esophageal hiatus. You can see this opening on many of our digestive system models in the laboratory. The gross anatomy of the esophagus is the wall of it is thick and composed of concentric tunics, or circular layers, that are continuous superiorly with those of the pharynx and inferiorly with the stomach. In the living adult human, the esophagus is about 25 centimeters long, and most of its length is within the thorax. Only the last 1.5 centimeters of the esophagus are located in the abdomen. And the empty esophagus is flattened. There is no continuously open lumen. It's only open when there's a passing bolus of food, which slightly expands the esophagus. Now let's talk about the histology, or microscopic anatomy. The mucosa is different from that of the abdominal GI, like I mentioned in the previous slide, or previous two slides. It's stratified squamous epithelium, and this is better suited to withstand the abrasions of the bolus that you just ate and swallowed as it moves through the esophagus. And because the esophagus does not absorb any nutrients, this thicker protective epithelium is warranted. The submucosa is thick and composed of elastic fibers. This allows for distension during swallowing, but also remember the recoilability is very important. The muscularis portion of the esophagus is composed of an inner circular layer and an outer longitudinal layer, like I demonstrated in the previous two slides of this segment. But the muscularis is unique in that it contains a blend of both skeletal and smooth muscles. The two layers of muscle in the superior one-third of the muscularis layer are skeletal, and that means you have voluntary control over them. But in the middle third, we have a mixture of skeletal and smooth, which means we're transitioning in the act of swallowing to more of an involuntary process. The lower third of the esophagus is only smooth muscle, which means the swallowing process and peristalsis at this point is involuntary. The smooth muscle is taking control of this. There are sphincters that are boundaries. There's the superior esophageal sphincter. It consists of that skeletal muscle, the superior third. This is where you have the voluntary control of this muscle. The sphincter would be closed during inhalation so that the air would enter your larynx instead of your esophagus. The inferior esophageal sphincter is now circular, smooth muscle. That means you don't have voluntary control over this with response to swallowing. And interestingly, you have to relax in some fashion both of these sphincters. Now you have voluntary control over the superior one when you vomit. If you do not relax your superior esophageal sphincter voluntarily while you're vomiting, you're basically going to have your vomitus come up, but it won't be coming out of your mouth, and that's called retching, and you end up swallowing that back down. Just a moment to highlight some pathophysiology of the esophagus. There is something called gastroesophageal reflux disease, or GERD. This is frequent gastric reflux. That means the stomach acids are getting through the lower esophageal sphincter. It's really not a true sphincter. It doesn't really block off the stomach contents from going back into the esophagus that well. But when the gastric reflux erodes the esophageal tissue, there's scar tissue that builds up in the esophagus, and this can lead to narrowing of that lumen. In more advanced cases, we see the simple columnar, or simple squamous, not simple squamous, but stratified squamous epithelium replaced by simple columnar epithelium. And this is known as metaplasia, when one tissue changes from one cell type to another. And this is called Barrett's esophagus, and there is an increased risk of cancer. We talk about GERD and Barrett esophagus quite a bit in my pathophysiology class. One way to treat this excessive acid production and reflux is by giving proton pump inhibitors known as omeprazole, so like Prilosec, and this will abate the acid secretions. There are other drugs that can be given, like histamine blockers, like Zantac, which is ranitidine, that can help limit the acid secretion as well. And you learn about all these drugs and their different receptors a lot in physiology. Another thing that can happen is through that lower esophageal sphincter and the esophageal hiatus, you can get some of the stomach and that lower esophageal area pulling through the esophageal hiatus, and some of the stomach is actually now in the thoracic cavity. Every once in a while we actually get a cat that has a hiatal hernia. That's what this means. Some of the stomach gets up into the thoracic region. And now this little hiatus, this band of skeletal muscle, the opening through the diaphragm, can act as a constrictor, and that's why some people feel like they have heartburn, they have some food stuck up there. And you mix that with some of the acid secretions, and it just makes them feel like there's a burning sensation. This is the slide that I was referring to earlier in our first segment when I said, depending on where you are in the GI tract, there are different actions for propulsion, moving your bolus and your chyme through the digestive tract. Now, deglutition is the act of swallowing. That's a voluntary process for the upper region of your esophagus, but then it becomes involuntary towards the esophageal phase, as it's called. That's the lower third, where it's smooth muscle that I was talking about. So swallowing is one mechanism of propulsion. And your book talks about the voluntary phase, where you're putting the bolus of food back on the posterior aspect of your tongue, and you push it posteriorly, and then you actually swallow. And then when the appearance of the bolus goes into the oropharynx, that's the beginning of the pharyngeal phase. And then eventually towards the bottom third of your esophagus, this action of pushing the bolus forward becomes involuntary. And basically, deglutition is really peristalsis, where you have a band of contraction behind the bolus, as shown in the top left picture here. And ahead of the bolus, there's something called reflexive relaxation, where the smooth muscle relaxes, and that helps to receive the bolus of food. In segmentation, up here in the top right picture, you're seeing local contraction from the muscularis externi. And it's sort of, I like to think of my hands on a piece of dough, and I'm squeezing here and there, mixing the components of my dough. That's segmentation. There can also be pendular motion, and by the way, peristalsis occurs in your esophagus, peristalsis occurs in your small intestine and large intestine. Segmentation occurs in the small intestine, and it increases mechanical breakdown of the food. And pendular motion is where the small intestine uses peristalsis, but it swings back and forth, and that helps with more mixing of the digestive enzymes with the chyme. The retropulsion action that is stated here is for the stomach. It's a churning-like activity. Have you ever noticed that when you eat a meal, your stomach gurgles more? And you might be embarrassed by this. You might lie and say, oh, I'm really hungry. No, you're not. Your stomach is loudest when you've eaten a meal, and that's because it's flipping the chyme around and mixing it. That's retropulsion. And if you really enhance that activity, you can get vomiting, where you are using peristalsis in reverse. Then in the large intestine, not shown here, there's an action called mass movement, and it's peristalsis gone crazy. You have 1 to 3 mass movements a day where the large intestine uses very strong contractile force to move the fecal material through the ascending colon, transverse, and descending, and into your sigmoid colon. You only need about 25% of your sigmoid colon occupied by fecal material, and you get the sensation that you need to empty your bowel. Whether or not you do or not, that's up to you. Okay, so those are the types of gut motility that are possible. How many of these exist from region to region, from the where you are in the GI tract? Well, that's where you need to stop and think. Would you have retropulsion in the small intestine? No, that's only in the stomach. Can you have pendular motion, back and forth action in the esophagus? No, that's not something normal. We don't have the food go back and up and down in our esophagus. Pendular motion occurs in the small intestine. I think you get the idea. Moving on to the stomach. The stomach is a muscular J-shaped sac, and it occupies the upper left quadrant of your abdomen. We learned about our quadrants in Unit 1. And it's immediately inferior to the diaphragm. In the stomach, we have both mechanical and chemical digestion. What's the mechanical? The retropulsion, the churning action, the mixing. The chemical digestion is going to come from an enzyme called pepsin that participates in protein digestion. I'll talk about that more in just a moment. The bolus is going to be mixed with all of these gastric secretions, and once the gastric secretions are released, we now call this chyme. Chyme has the consistency of a paste-like soup. Ugh. The stomach facilitates mechanical digestion, like I said, by contractions of the muscularis externi. And by the way, there are 3 layers now of the muscularis externi, which I will talk about, not just 2. And again, the stomach is going to facilitate chemical digestion through its gastric secretions. I will talk about that extensively in just a moment. The gross anatomy, well, let's talk about the regions of the stomach. There's the cardia, or cardiac region. This is the small, narrow, superior entryway into the stomach lumen from the esophagus, shown here in the top of this picture. The internal opening where the cardia meets the esophagus is called the cardiac orifice. The fundus is the dome-shaped region, lateral and superior to the esophageal connection with the stomach. Its superior surface contacts the diaphragm, the underside of the diaphragm. The body is the largest region of the stomach. It is inferior to the cardiac orifice and the fundus. The pylorus, or gatekeeper, is a narrow, medially directed funnel-shaped pouch that forms the terminal region of the stomach. The pylorus is divided into 2 parts. The pyloric antrum, the more expanded region near the body of the stomach, and the pyloric canal. This is a more narrow region that attaches to the duodenum. Its opening with the duodenum of the small intestine is called the pyloric orifice. Surrounding this pyloric orifice is a thick ring of circular smooth muscle called the pyloric sphincter. The pyloric sphincter regulates the entrance of chyme into the small intestine by closing upon sympathetic innervation and opening upon parasympathetic innervation. We know that, right? More gut motility, parasympathetic division has more control. There are also curves to the stomach. There's an inferior convex curve, and that's the border of the stomach. The inferior convex border is the greater curvature, whereas the superior concave, or indented region, is the lesser curvature. That would face towards the liver. And remember, the greater omentum attaches to the greater curvature edge of the stomach, and the lesser omentum extends from the lesser curvature to the liver. Inside the stomach, and you will see this in the cats, there are numerous gastric folds, or rugae. These gastric folds, which are observed only when the stomach is empty, allow the stomach to expand greatly when it fills, and then return to its normal J shape when it empties. The muscularis externa, like I said, there are 3 layers to the muscularis externa. We have the outer longitudinal layer, we have a middle layer, that's the circular layer, and the most internal layer is the oblique layer. That's the most internal layer. This is very unique, 3 layers to the muscularis externi. Now I want to tell you about the stomach and digestion and absorption of the stomach. Let's see, how do I want to approach this? Before I discuss the actual secretions and absorption of the stomach, let me first describe the motor functions of the stomach a little bit more. The motor functions of the stomach are threefold. 1, storage of food until the food can be processed in the duodenum. It's not really food, it's chyme. Mixing of your food with gastric secretions to make this chyme. And then 3, emptying of the chyme into the small intestine at a rate suitable for proper digestion and absorption by the small intestine. The stomach relaxes when food, the bolus that you swallowed, enters, and it can accommodate approximately 1.5 liters of food when completely relaxed. Each time a peristaltic wave passes over the antrum toward the pylorus, the pyloric muscle contracts, which further impedes emptying through the pylorus, and the food in the antrum is squirted backward toward the body of the stomach for more churning and mixing of food with gastric juices. This is the retropulsion I was telling you about. I'm going to talk about the secretions of the stomach only briefly here, but we'll expand more in the next few slides. You will learn in more detail that the stomach has parietal cells that secrete hydrochloric acid and intrinsic factor, and you will also learn in these next few slides that there are chief cells that secrete pepsinogen. We'll also learn about the cells that release gastrin and the cells that release the mucin that turns into mucus. Very little absorption occurs across the stomach epithelium due to the gastric mucosal barrier, which is composed of the mucus secreted by mucus cells and the tight junctions in between the gastric epithelial cells. Some substances are able to be absorbed, substances like aspirin or other NSAIDs, and alcohol. Anything that's lipid soluble that can get through the tight cell membranes can be absorbed into the bloodstream found in the submucosa. The stomach has 2 important types of tubular glands. Your textbook only shows 1. There are 2 tubular glands. First of all, please stop and focus on this picture. There's a difference between a gastric pit and the tubular glands that I'm referring to. Let's first address the gastric pits. The stomach mucosa has many indented, numerous depressions called gastric pits. At the base of each pit are openings of several branched tubular glands. There are 2 kinds of tubular glands: gastric glands that your textbook talks about, but also pyloric glands. And the cells you find there are different. So I'm using the picture from your textbook, but you need to follow my voice and these PowerPoints and know which cells you find at each gland. 80% of your tubular glands are called gastric glands. Look at the picture in the top right here where I'm showing the stomach and it's highlighted in gray. That would be your fundus, your cardia region, and your body. Here you find gastric glands, and there are 3 types of cells. Of course, we find the mucus cells, whether it be the surface mucus cell or the neck cell, it doesn't matter. We find mucus cells, and they release the mucin, which mixes with water to form mucus. And this helps protect the simple columnar cells that are facing your chyme, where all of your acid and digestive enzymes are. You know, the stomach is going to, I'm going to show you, it's going to release an enzyme that digests proteins. Hello, cells are filled with proteins. You don't want to digest yourself. So this mucus slathers and protects the epithelium so that you do not digest yourself. All right, so I've now covered the mucus cells, but the gastric glands have 2 other types of cells as well. They have the peptic or chief cells. These are going to release pepsinogen. And you also have parietal or oxyntic cells. I'm just going to call them parietal cells. And these secrete hydrochloric acid and a substance called intrinsic factor. The pyloric glands I will talk more on the next slide. They are located in the antrum primarily, and they secrete mainly mucus for protection of the pyloric mucosa, and very importantly, the hormone gastrin. There's not really a ton of pepsinogen because there aren't many chief cells there, so I'll talk more about the pyloric glands in a moment. Let's focus on the parietal cells shown here in this gastric gland. they secrete hydrochloric acid, and hydrochloric acid is as necessary as pepsin for protein digestion in the stomach. The pepsinogens have no digestive activity when they are first secreted. As soon as they come into contact with hydrochloric acid released by the parietal cells, then the pepsinogen is changed to an active form called pepsin. Parietal cells also secrete intrinsic factor, which is essential for the absorption of vitamin B12 in the ileum of the small intestine. Without vitamin B12, you will not make your red blood cells appropriately, and this can lead to a condition called megaloblastic anemia. When the acid-producing cells of the stomach are destroyed, which frequently occurs with chronic gastritis, the person develops not only achlorhydria, which means lack of being able to produce the acid, but often pernicious anemia or megaloblastic anemia that I just referred to, and this leads to failure of red blood cells maturing appropriately. The other or third cell that we find here are chief cells. They're called chief cells because they are very numerous. They release pepsinogen, which is an enzyme, and anything that is an ogen or a zymogen means it's an enzyme released in an inactive form. Hydrochloric acid will create a very low pH in the stomach, and that is what is needed to activate pepsinogen. That should be really strange to you. Most of your enzymes are active at a normal body pH, which is 7.40. The stomach acidity can drop to 2, and that's what's required to activate this particular enzyme, pepsinogen, into pepsin. Pepsin breaks down proteins into smaller peptides, but it is the hydrochloric acid that first denatures the proteins. That means it merely unravels them from their quaternary and tertiary and secondary structures down to their primary structure. That means the pepsin now has an easier time to break apart the peptide bonds in between the adjacent amino acids. You should be hearing that protein digestion begins in the stomach with the action of pepsin. Pepsinogen is secreted by chief cells and has no enzymatic activity until exposed to the hydrochloric acid secreted by the parietal cells. The hydrochloric acid secreted can have a pH of as low as 0.8, but by the time it mixes with the stomach contents, its pH climbs to a bit higher, between 2 to 3.0, which happens to be the most optimal pH range for pepsin activity. Pepsin will lose its enzymatic optimal activity at pHs above 5. Pepsin only initiates the protein digestion, usually providing only 10 to 20% of total protein digestion. The majority of protein digestion is going to occur in the duodenum through enzymes secreted by the pancreas. These will be discussed later. The other type of gland that I want to talk about is the pyloric gland. The pyloric gland has two primary cell types. Of course, there are the mucus cells that are producing the bicarbonate-rich mucus, which will protect the mucosa of the stomach from it digesting itself. But deep at the bottom of the pits, shown here in a kind of a turquoise color, are enteroendocrine cells. These are cells that are endocrine glands, single-cell endocrine glands. Entero means gut, endocrine cell of the gut. That's what their name tells you. These cells release a hormone called gastrin. When gastrin is released into the blood, it in turn circulates through your body only to target the parietal cells of the gastric glands that we just discussed. The parietal cells in turn will release even more hydrochloric acid and intrinsic factor because of gastrin targeting them. The question is on this slide, if we can eat tripe, which is stomach from another animal, why don't we digest our own stomach? That's because those mucus cells release mucus. The mucus is alkaline, that means bicarbonate rich. It has a high pH, and that will immediately neutralize the stomach acid when it tries to come into contact with the simple columnar cells. Additionally, the simple columnar cells have very tight junctions in between them. This inhibits the gastric juice and enzymes and hydrochloric acid from backflowing or leaking and digesting your own cells of your stomach. Additionally, as the cells succumb to some of this chemical digestion, the simple columnar epithelial cells are replaced roughly every 3 to 6 days. So we have a lot of intrinsic protective barriers against our own digestive enzymes that are being released into the stomach. That's why, under normal healthy conditions, we don't digest our own stomach. But if these protective barriers break down, then you will digest your own gastric lining, and this will lead to gastric ulcers, or sometimes even these ulcers can present in the duodenum, as shown in this top picture. I will talk more about ulcers, peptic ulcers, and what that means, gastric ulcers versus duodenal ulcers. Why did they start in the first place? One is from a lack of protection. The other is from excessive acid secretion. I will talk about these on the last slide of our lecture. This concludes our second installment of the digestive system. Whew. We have 20 more slides to go.